Yes, thanks. Thanks for the invitation. Thanks, Pascal. So it's my pleasure. Please, I know like during the afternoon time like that after lunch, it's always, do not hesitate to stretch because, you know, like I know like you could, some of you doing Tai Chi, so you could do the tiger extensions if you want, so do not hesitate to activate. So I try to stretch a little bit you guys about vaccines development. I've been even told to do that start to finish vision, but you know, it's a lot of things to take care of. I could see over here, especially in Seoul and with Institut Pasteur, you're a little bit more at the preclinical level innovation and of course clinical as well, but not under GMP. So I'm gonna pass certain slides that are a little bit more about, you know, the GMP process, but do not hesitate to talk to me later on, you know, if you want to know more about CMC innovation and things like that. So just I'll talk about all of these things rapidly, but just to let you know, in our case at Sanofi Vaccines, of course, we're working closely with Institut Pasteur all over the world, in the network, but especially in Institut Pasteur Paris. We're located in different countries. As you could see, in my case, I'm coming from France, at the Marseille-l'Étoile, Lyon area, the legacy of Mérieux, which was a student of Pasteur in Lyon. And then, of course, we have our R&D site as well in the US. And as you could see in Korea as well, we have like certain collaborations, especially even at CMC level or GMP level with SK Bioscience. We have our pneumoconjugate vaccines under phase 3 trials at this time. So we're really excited to work with Korean biopharma. So I think for us, as I said, SK, it's really important. We're working on the pneumoconjugate. We're also working with Sanofi Pharma a little bit more on chronic disorder or neural disease, with other biotechs such as ABL Bio on Parkinson's disease, and even others, you know, in terms of chronic disorders, as you could see, and we have certain partnership with Seoul Center. So we have a hub over here also at Sanofi in Seoul, you know, taking care of the medical affair for Sanofi. And of course, for us, as you could see, respiratory, dermatology, especially here, it's really key, you know, and for Sanofi. And apparently, there's also like different type of collaboration agreement that has been done. And we're looking forward to have a collaboration agreement with Institut Pasteur Korea as well, not just the one with Paris we have with Pascal. So just to let you know, in terms of platform, we're not a one-size-fits-all company. You know, we're not like Moderna, just taking care of the mRNA. We believe, you know, like in the human and the biology is complex. We need different modality depending the target, depending the disease. So we need to have expertise in different things. And this is a little bit like an overview. I don't want to go too much in details. All of the platform we have at Sanofi and we're working on, as you could see, of course, there's more and more biological therapies using antibody or targeting system, including even within the mRNA, such as in vivo CAR-T mRNA, that is ongoing at this time a lot. The lipid nanoparticle that we acquire from Translate Bio, which now is competing, you know, like Moderna and BioNTech, Pfizer on this. And so we gain all of the experience on this. And so it's really important to have good science and have a good relationship with you guys, like the expert in the field, in the early innovation. So that's why I'm here today. I'm traveling all around the world to talk to rising star and established star, and we have a system, you know, to do that. And just to let you know, this is our pipeline at this time at clinical level, phase 1, phase 2, phase 3, as you could see. So you could see there's certain target that or attack it, or we are working and pursuing it, but you could see there's other infectious disease target that are or less pursuing at this time because there's a need. So they are at the preclinical space. I don't have a slide presenting all of what we do at preclinical stage. You will see later on. But just bear that in mind in terms of the target. With Institut Pasteur Paris, we have a good relationship, and I really like, you know, work with Institut Pasteur Paris, especially like the dairy organization and the relationship we have. So I started to be the coordinator of the science with Pasteur 4 years ago, and we built over 12 contracts, including, you know, researchers that are here today on different type of aspects, as you could see in terms of innovation, coming from even human profiling to even microbiome evaluation. Like we try to see what's the impact of microbiome change on vaccines responsiveness, on structural biology such as cryo-EM, chlamydia, so STIs, sexually transmitted disease, we need to understand a little bit more. And as you could see also in terms of mRNA, it's really key. In the last year, we've been working on 4 to 5 contracts on mRNA, including the biology, the cell biology on mRNA biology and mRNA vaccines, including also cell-based assay to do screening with Dr. Kumarova, such as all of this RLR and PKR binding assay, and also others such as immunology as well with the late Philippe Bousseau, but now taken care now by Dr. Grandjean as well. And of course, we have Chris Zimmer that's going to talk also. We're working a lot with the our epidemiology team on machine learning and AI as well, like to understand a little bit more infectious disease and also vaccines intake. So, of course, like we're working in different type of platform. As you could see, this is the Big Six platform. When I go to university, sometimes I need to do a course to explain all of the Big Six, the 6 major vaccines platform exist in the world. So, as you could see, mRNA are part of the genetic vaccines platform, even like at the time with DNA vaccines, it was existing. So just in terms of flu, so there's not a one-size-fits-all. So it's important for biopharma to have, depending the needs, such as the immunology needs in the world, such as the immunosenescence. You might need a type of vaccines at elderly stage, which you don't need, at young stage. So that's why in our case, we invest in different type of vaccines platform before I present that. But as you could see over here, the split vaccines platform is one of the most important one, and we're still working on this. But there's also other type of subunit vaccines we're working, especially in terms of protein recombinant protein as well, without egg base, because there's certain people that they are allergic to eggs extract, so we need also to develop other type of vaccines. So that's why we have a pipeline with different type of vaccines for flu, such as Vaxigrip, which is the split flu, compared to Flublok, which is the cell culture baculovirus expression system flu HA, and also like the Influelda now that has been shown that a high dose of flu vaccines is needed to overcome the immunosenescence a little bit more. And of course, we're working on next generation flu, as you could see as well. So in terms of vaccines platform, also in the polysaccharide conjugated vaccines platform, we're working closely with Korea Biopharma, such as SK Bioscience, as you could see here, in terms of the polyconjugate vaccines, like using a different type of carrier proteins, but where we conjugate the polysaccharide to make them immunogenic. And the phase 3, it's ongoing at this time, especially on the 21st valency with SK Bioscience. And in terms of in terms of GMP also, and also needs in the world, as you might know, one of the best vaccines with single injection is the live attenuated vaccines for yellow fever, which is based on a culture in eggs, embryonic eggs, first, the YFVAX or Stamaril vaccines, which exists since over 40 years and more. And now we're working on the second generation vaccine, especially to avoid, this egg extract and have a highly purified recombinant type of vaccines, or recombinant, on protein-based vaccines. And we're working on the second generation on Vero cells, and that's really, of course, in terms of innovation, in terms of GMP and fermentation, it's also needed, and that's why that famous end-to-end need to take into account also the CMC and fermentation step. Another platform that it's really important for company, we saw that in the last year, especially with SARS-CoV-2 and also RSV. I would say before SARS-CoV-2, it's the importance to lock antigens, especially at the pre-fusion state. In virology, often we need to attack a virus before it attaching, like the epithelial cells, such as for respiratory virus there. So we need to kind of fix with different type of technology, a pre-fusion state in our antigens design. And now we're working with VisBio to have that famous molecular clamp, like it's an improvement of the T4 fold-on that was used from the COVID vaccines, subunit COVID vaccines, to really have clear a trimer that's going to be stayed there as a trimeric form, and then we'll be able to get an immune response against a trimeric form, such as for spike, or such as also for RSV, and also HMPV or parainfluenza virus. So if you're working this type of target, please come to us, and we could have certain agreement, with Institut Pasteur as well, on this type of antigens design innovation. So as you know, that famous end-to-end, it takes time to make vaccines to human, to go even at registration state. This is just an example from vaccination. And even I could tell you, often it's more than that length of development, especially at preclinical phase. Just RSV vaccines, an example, where the preclinical phase is ongoing since 20 years. Well, of course, now we have vaccines, but it took over 20 years of preclinical phase to make this type of vaccines. So it takes time. We need to be patient. So we need to have a strategy. In Korea, that's the same thing, I believe. If you want to build this type of strategy of innovation, you have to think about all of the value chain from preclinical, early innovation and publication to until the end at GMP production of vaccines. So that's why in our case, we really invest to have that end-to-end type of platform within Sanofi in terms of all of the big 6 platform, of course, but also in terms of analytical and assay, automation as well. I was talking to Steve. It's really important to have a good automation now, and even miniaturization and automation adapted to miniaturization, such as microfluidic. Manufacturing, same thing as well. We could not have just one factory per vaccines. Now the evolutive vaccine facility, where 1 month we could do a batch of GMP production vaccines, and after that we could change even fermenters, such as a disposable fermenter, to do another type of vaccines. So it's possible now, and that's what we need to work on. So this is also like a little bit like an overview, of course, of the expertise you need to have, when you're starting to work on end-to-end process of vaccines production. As you could see, of course, there's multiple steps in terms of process development, such as like the virus or bacterial expression system, or even like producing, like the genetic material. And after that, there's a lot of steps in terms of valency assembly, formulation, as you could see, filling, and of course, as well, like all of this cold chain respect, as well as the packaging, and at the end batch release. So all of this, as you could see, each vaccines contain from 1 to 9 antigens often, and production, and testing take 6 to 36 months per vaccine for release. So just like an example of what you all need, to get in terms of the big 4-stage of the end-to-end process, including in terms of the R&D at the beginning, characterization, to of course, when you start to work under GMP or GLP environment, and of course, even at the end in terms of the characterization package, which the regulatory agency, such as the MFDS here in Korea, well, needs also to have. So there's certain guidelines to respect. And of course, like when the new platform came out, such as mRNA, that was also the same thing. Of course, when you come after you have that burden you have that burden of having more and more tests for the release of the vaccines, which is good because we ensure the quality. And you could see, from the first generation of mRNA vaccines, especially compared to the latest generation of mRNA vaccines, there's a lot of improvement that have been done, such as what you just discussed, Dr. Kim, about, like the polyadenylation evaluation as well. There was a lot of heterogeneity even in the final product for SARS-CoV-2, which so perhaps was one of the explanations also of reactogenicity of this type of vaccines. So that's why we really need innovators such as you guys that gonna working also to try to get like this steady, little bit more way of analyzing mRNA. And of course, you have to keep in mind for having that end-to-end, you need a manufacturability, you need stability, and also deviceability. It's not just about making a vaccine in a vial. Often, you know, we need to also to think about the syringe process. Will it be a dual syringe process, especially when you have a subunit vaccines where you need an adjuvant or not? Is it freeze-drying? Is it liquid? As this time, as you know, mRNA needs to be liquid, and there's some stability issue, like the cold chain. We could not go at room temperature at this time with this type of vaccine. So there's this type of things that you need to analyze for that end-to-end process. So I'm going to take an example of like a live example of end-to-end process as we've done in the last year on mRNA vaccines. As you know, we have over 12 clinical trials using mRNA vaccines at this time, and especially just in terms of even platform testing with certain antigens such as flu that we decide to test as a platform to screen and on this mRNA vaccine. So as I mentioned before, there's a lot of things that needs to be taken into account, such as, of course, the region beside the coding region. It's not just about having, you know, a start codon to a stop codon and a Kozak sequence. You need also to think about the UTR, as we mentioned, and the polyadenylation. So all of this need to take in account. And the beauty of mRNA technology, it's true, it's not just about making a protein for vaccines or immune response, but you could also make gene therapy, and that was the case, even for Moderna at the beginning. It was a little bit more for gene therapy purpose, and also for antibody production as well, and even others such as nuclease. So the CRISPR-Cas system could also be done, using mRNA coding sequences. So in terms of, when you prepare for RNA vaccines, it's not just about the mRNA, but it's also about the lipid nanoparticle. I don't want to go too much in details, but as you know, the lipid nanoparticles, such as Peter Collis, that really with Aquitas and other company, that they really been pillar, like the methylpseudouridine, even alone, without not being able to be successful, without having that LNPs that protect. So it's kind of that liposome even was really super key, especially the cationic liposome development was super key. And that's why, this Trojan horse system was really important. So you have to take into account, it's not so easy to make mRNA vaccines, especially I would say in terms of the LNP evaluation and usage, because there's different type of lipid. Of course, you have the cationic lipid, but you have also the helper lipid that you need to think about. Would it be like DSPC or would it be DOPI? As you might know, BioNTech versus Moderna, if different type of helper lipid, and same thing in our case as well. Also, what type of cholesterol do you need, especially if we have seen this type of cholesterol before? Is there an impact, in terms of efficacy of this cholesterol, and also the pegylation of the lipid as well, which there's certain innovation that could be done there, and where we could tweak with the chemists all of this type of modification as well. So you need to screen a little bit around these 4 type of lipid, and that's what we've done in the last 6 years in our case, from preclinical screening, such as in vitro expression in certain cell line, as we saw, like with the 3' UTR and polyadenylation, and until also the animal models, and not just in mice, but also in primate, and also even going in human if you could. So we've been able to build like a machine learning system and a huge database system that take into account all of these things, including even now like the testing that we developed with Institut Pasteur Paris, such as there's RIG-I or PKR activation assay and others. So all of this are taken account in our big machine learning system. And we publish about this. You could see like the publication we made, but this is just an example that has been also put in the publication. So when we discussed with Steve at IPK, that's the same thing. We need this automation system, even at a gene expression level, with the Operetta Perkin-Elmer's system in 96 well plate, and even in the 384 well plate system, where you could evaluate over 500 mRNA sequence at the same day. And after that, of course, you need to analyze the expression as we saw, like with this tail difference as well. We do the same in terms of the different type of sequence, in terms of the codon optimization as well. So that's what we've been able to do, like having like a screening system all integrated together, including this translation assay and this expression assay overall. And one of the things that was also shown, in methylpseudouridine, and I was talking yesterday to Dr. K. Zhang about all of this importance of working on translation and ribosome. And one of the things that was done also, not just with Quérico, but also with Sonnenberg and Anne-Claude Gingras in Canada, it was that ribosome stalling process that has been shown. The methylpseudouridine, the reason why you get a nice protein expression is because it seems that the ribosome is stalling a little bit more, so you have a high density of ribosome and you could increase the translation. So it's not just about also having less PKR activated by methylpseudouridine, but it's also about having all of this translation mechanism that seems to be increased. And we're looking forward for other modifications such as K5 and K7. So at the end, same thing, we do the same with the LNP, especially with the different type of PEG and different type of cationic lipid. The chemist needs to work on all of this type of lipid nanoparticle as well. And you compare, of course, with the Moderna one with MC3 or SM102, coming from Aquitas. And same thing, like we need to see, do we have better LNP that could help, in terms of a translation and even in vivo as well, where we could do also that type of test. One thing that it's a need in the field, it's also the stability of mRNA vaccines. So could we work a little bit on a stabilized prototype or even a lyophilized LNPs? And at preclinical level at this time, that's something that seems to be possible now. And we'll see in the future, we're evaluating at clinical level also new LNPs that seems to be stable for a longer time, as you could see at 4 degrees compared to the standard at this time where after 3 months, you need to do another batch of mRNA. So that's what our ecosystem in terms of end-to-end, especially with mRNA. We could do a lot of things, not just about vaccination. We're working at this time on mucosal delivery of LNP mRNA vaccines. We're working also on oncology, such as CAR-T, especially the in vivo CAR-T, where you inject an LNP that we're going to code, for a T cells receptor, a TCR, and we're hoping that that LNP mRNA will go to the T cells to have an anti-cancer, of course, when you want to get, a CTL response. And same thing in terms of Cas9 system or others on gene editing and even other type of gene therapy technology. So overall, that's what we have to say in terms of the platform. There's still something to overcome, especially with university. Such all of this immunity question, could we get also like a better alternative route of vaccination? So that's a major question. Of course, we want to work with you guys in Korea for this, and of course the MR vaccine has been like one very good example of that end-to-end. And as I said before, there's no one-size-fits-all technology, all of this. So that's a little bit what I want to, and of course it's important to have a broad portfolio of vaccines platform because we could not rely on a single platform. We need to take into account the CMC and also like the machine learning and AI analysis, especially when you start to screen like that. And of course, we want to go in future with next generation vaccines, even chronic disorder. I want to take the 1.35 minutes remaining to talk about a possibility of grant you could get, especially from if you're a biotech or if you're an academic, such as Institut Pasteur Korea, where we're working now with the external innovation team because Institut Pasteur Paris could apply to this type of grant. So I'm going to talk about the grant system that we would like to implement also in Korea. So where the researcher will be able to apply for this. Just to, before, sorry, I went too fast. The biopharma company like us, we realize often when the innovator were coming to us, the proof of concept were not fully ready. Like say, like there were certain control lacking, the benchmark were not there, so we could not make a decision, are we doing like a license option on that technology or not? So often, The researchers were coming back to the lab, and sometimes they didn't have the expertise or even the money to do it. So now we have a system where the biopharma, such as Sanofi, we could say, okay, we're going to invest on the proof of concept. We even don't want the IP. The IP, the intellectual property, the patent, going to remain to you. We pay for the research over there, but at least we're going to get a first sight, a first vision of the technology, and we're going to work with you, to do that proof of concept. So that's a little bit what we try to do in terms, because of course we have other type of system after that when the technology are mature, such as the Sanofi venture system and business development deal, which was the traditional way, but now the world is shifting to the left pre-seed funding program. So what about the pre-seed funding program? In our case at vaccines, this is the scope. It's really clear we cannot work on everything. So this is the scope we have at this time in terms of where we're looking for innovation. So do not hesitate to take the picture of that slide if you want, and I think you're going to get the slide as well. So we put that in our website. Every year, please, came out with proposal around this area. We will be happy to work with you. And that's what we do with Institut Pasteur Paris. And as you could see, also, there's other things that are super interesting in terms of new target. As you could see, the famous are not to crack target. And even I could tell you now, we're not just working on traditional vaccines, but also antimicrobial, so antibacteria or antiviral, especially if there's some biological therapies, let's say using, instead of just small drug molecules, using like targeting system, let's say with an antibody. That's something we're really looking at it. I don't want to go too much in details, but this is like our in scope at this time. And if you're working on this type of field, it could be very good to apply to this 3 type of grant called I-Award, Idea Tech. And we launched in the last year because this proof of concept of just 1 year might not have been enough funding and timing to do it. So we create another type of a grant system which called INEX. And so we're doing this type of research for call, RFP, every year now. And I'm hoping that especially the INEX are going to be available in 2027 for you guys. So we're going to work. And this is like the website at this time, for the 2026 proposal in our website where you could apply for this type of grant, which are, just 5 pages of grant. And in our case, we're evaluating it, and we try to make a contract within 6 months after the full application of this grant. And this is also like in terms of the iNext, where this year we were targeting a little bit more moonshot approach, such as longevity and also targeting systems, especially intracellular targeting system, which, as you could see, beside LNPs, there's a need at this time, to make enter biodrug. So that's it. I don't want to go too much in detail, so we thank you, and I'm looking forward to talk to you after the session. Thank you.